In the last discussion, we established this idea of multiple sequence alignment as an NP-complete problem, which means that as the number of sequences that we're evaluating begins to grow, soon the number of possible alignments gets so big that we cannot get a optimal solution, at least to be 100% sure that we have the optimal solution, because there are just too many solutions to evaluate. And so we're left to algorithms or search strategies. Now, before we move on, I just want to make 2 points about the reading. Number one is that we don't have a textbook for this class, and so that is a little bit tricky sometimes. There's really not a good textbook that covers everything we wanted to do, and so I'd have to have you buy multiple textbooks. And so I think readings from the literature are a better solution. But number two, the second point, what that means is that some of the papers are going to be quite technical and are going to go into depths that we don't really need to get into. So as you're reading these papers, if you're getting overwhelmed, don't freak out too much, okay? Skim over them, review them. If you're getting into it, read them in a little bit more depth. Then come to the lectures, go over what the main material we're going to do in the lectures, and then you can go back to the papers if you need to review. But you don't feel like you need to master or certainly memorize all of the materials in the paper. So what we're going to do with multiple sequence alignment, because it's a very complex topic, it's a very technical topic, we could spend an entire semester on it, and there's still lots of ongoing research that is continuing to develop it. So our goal here is not to give you an exhaustive, an exhaustive discussion about multiple sequence alignment, because that would take weeks and weeks. We're just going to give you a big picture overview and hit some of the highlights. Okay, so because a multiple sequence alignment is attempting to make statements about the history of sequences, right? Homology statements saying that this T, this C, and this T all come from a common ancestral nucleotide in some ancient ancestor. And because that is a historical statement, we never know 100% that we can have, quote unquote, a true alignment. In fact, there probably is no really such thing as a true alignment unless we could somehow invent a time machine. And even then, you would need to get into the genetics and sequence the DNA of organisms as speciation occurred, as fixation processes changed one common nucleotide residue due to a different one in the population. And so in reality, we can never know for sure. In a way, it's a little bit like reconstructing history of the human species, right? So if we know something about, let's say, World War II, we have evidence, we have multiple witnesses that might give us good indication that we have a good idea of something that happened. But again, we never know for sure because it's a historical reconstruction of a past event. So in the same way, where our idea is to get good, workable, usable alignments that give us a good estimate, maybe even the best estimate of how those sequences are connected to each other, but maybe there's no idea about this true alignment. So maybe we shouldn't worry so much about getting the global optimum as long as we have a good approximation or workable alignment. Now, to do that, there are two main ways that have been used, and then kind of a new, or maybe even you could consider it a subcategory of the third way that we are going to talk about. Okay, so a progressive alignment basically breaks the problem of multiple sequence alignment down into reasonable subcategories, and then we can perform each subcategory. And most of these progressive alignments use a guide tree. And so this guide tree can be built by simply doing pairwise alignments and finding which sequences have the shortest distance, which ones are the most similar. And then we align the most similar sequences together first. So notice here there's only one difference, so we align those first, boom, we're done. We then take a version of this one. Maybe we fuse them together and put a little note on this, what is it, the 7th base pair sequence there, that can be either or. And we compare it to all the other sequences and find which of the next sequences is most similar. We get a score. And if two sequences stick together that with a lower score than this amalgamation sequences sticks to anyone's, then we do those first. So we would, in this case, I guess they did these ones first, then they did these two, then they align this third one, and then they align the fourth one. And so we can progress step by step by step and do a pairwise alignment at each step. And so these are usually rather quick, although sometimes evaluating lots of sequences and their distances can be tricky. But this is a fairly simplistic, in most cases, particularly if the sequences are not too divergent, yields pretty good alignments. There are many versions that do this. Clustal is maybe the most widely used of all of these, but you might come across some of these other versions. So progressive alignments break the problem down into subcategories, and then they go ahead and solve it. They're rather quick. They give pretty good alignments, especially for sequences with fairly low divergence. But the downside of this is with progressive alignments, you can get locked into one pattern. And so it's not attempting to find a global overall best optimal solution, even if we can't search every solution. It's not attempting to give you a best fit. It's just saying, well, let's break this down into a reasonable way. Do a heuristic search where you do A, B, C, and D. We do those steps one by one by one, and at the very end, we'll get an alignment. Whether or not it's the best one, it will probably be a pretty good one. Now, an alternative to a progressive alignment is an iterative alignment. Now, again, there are multiple ones. You might be familiar with some of these, or you might come across some of these. These are commonly used one. MAFFT is perhaps the most commonly used one for big data sets. Muscle is also a fairly common one, and that's an algorithm that's available in the Mega software program. Now, with an iterative alignment, it's attempt to search all the possible alignments and do that over and over and over again in some way that we can be somewhat certain, or maybe at least have a fairly good chance of finding something close to the optimal alignment. Now, to do that, we need some sort of a criterion, a score. And so basically what we do is we take the unaligned sequence, we do a very quick and dirty alignment, the best we can, right? Not one that's going to take a long time, maybe a very quick progressive type alignment. And we use that to generate a tree showing how those sequences are related. This tree then goes into allowing us to do a little bit of a better alignment, and then we reconstruct the matrix. We get another tree. We use that tree to reconstruct the alignment, and we do this over and over and over again. So we're going from alignment to tree back to alignment. And every time we do this, we get a score out. And that score becomes our criterion for whether or not the alignment is better or worse than the alignment that we had previous. And we do this over and over and over again until our alignments scores are no longer becoming any better. And so we could do multiple versions of that. Now, with an iterative alignment, we have more chances because we're redoing it and reevaluating, allowing the original alignment to then be flexible. We don't get locked into any one category. Now, the downside of this is that it is very computationally expensive. It can take lots and lots of time and effort. Another thing that we can do is we can do multiple versions of this. So if I start in an alignment and I get some sort of a plateau where I'm not getting any better scores, and then I redo it, and if I get to that same plateau where I'm getting the same alignments or roughly the same alignments with the same scores, and I do that over and over and over again, starting with a random alignment, then I can be somewhat certain that I'm finding the best tree overall. And so this is a little bit more of a common method to use because we're more of an optimality criterion. And we're going to talk in detail what we mean by heuristic searches versus optimality criteria, particularly in reference to phylogenetic inference. And we'll do that at a later discussion. But for now, just know that an optimality criteria tends to try and find the best overall solution to a problem, in this case, the best overall alignment, based on some objective score. And then in an iterative version, we go back and do it over and over and over again and tweak it, move it around, allow it to change a little bit. And so therefore, we have more of a surety that we found something close to the best overall score. Now, two downsides to this. One, I've already mentioned that it's very computationally intensive, takes lots of computer resources, and might take a long time. The second one is that this is sensitive to whatever parameters we are using to measure our optimal score. And so there really is no 100% way to come up with the best overall parameters. And so sometimes we may even need to make this a little bit more computationally intense by tweaking the parameters of how we are estimating what this best alignment is. Okay, so that's a big picture overview of iterative alignments. They are basically optimization solutions. They're more computationally intensive, but have a better chance of giving us a better overall view. And when I talked about Cobalt, which was the multiple sequence alignment tool that uses the BLAST algorithm, that is one of these iterative alignments. Okay, it falls into that category. Okay. Now again, we never know that we have the correct alignment or the true alignment, because in reality there's really probably no such thing as a true alignment, because we can't go back in history and know for sure that that's how those sequences evolved and that's how those homology statements are best put together. Okay. But when we have very similar sequences, especially as we get closer and closer to that 100%, this is easier. And so whether we do a progressive alignment or an iterative alignment, if the sequences are not too divergent, we're probably going to get a very similar or maybe even identical alignment out of that, and it probably doesn't really matter. And so maybe we choose the faster method if we don't have very divergent sequences. If the sequences get more divergent, then we might want to invest more time and spend more resources searching what is going to be the best overall alignment. And a more recent way that people have done this is using a genetic algorithm, which really isn't truly either a progressive or an iterative alignment. It falls into this category of what we're going to call a stochastic approach. And a stochastic approach uses random perturbation at each cycle and then redoes it. And these genetic algorithms are just one version, but a commonly implemented one, of a stochastic approach. So as long as you know that a genetic algorithm falls into this category of stochastic approaches and that there are others, that's as detailed as we're going to get. And this is a huge topic of research for bioinformatics and gets really in-depth and involves mathematics and theory and a lot of other things. So you're going to see things like this. We're not going to freak out or worry about what each of those different variables means and how we can implement them mathematically to find it. That's beyond what we need for this class. But the genetic algorithm implements a stochastic approach that is somewhat similar to the process of evolution. That's why we call it a genetic algorithm. And there are different components. We break these down into subcategories, and then we randomly mix them together. So we might take part of the alignment, break them into subcategories, and then mix them back together and see if we get a better score. So that mimics to some extent things like crossing over or random assortment. We can also perturb or change the alignment slightly in a random way, which might mimic a mutation or some process of genetic drift. And then we get a subcategory of alignments out of that that are either better or worse than the previous alignment. If they're worse, we discard them, and that mimics natural selection. If they're better, we keep them, and we use those as the foundation for our next round of alignments. And we do that over and over and over again, always keeping a view on that alignment score. And if it's better, then we continue the process. If we get to a point where no matter what we do, the scores are not getting better, then we realize that we've probably optimized it as best we can using this algorithm, and we don't do any more searches. And so again, we can do lots and lots of searches. And these genetic algorithms, again, the downside is they're fairly computationally intense. But the stochastic approach may do an even better job of doing an overall search of the possible solutions. And so we can be a little bit more certain, if we invest the resources, that we might have found a best overall solution. The downside of this is very similar to the iterative, sorry, that's tricky to say. It's very similar to the iterative approach, meaning that if our way that we are measuring the very best over alignment is not very good, then no matter how much we do this, we're not going to have a best overall solution. But again, remember, we can never be 100% certain. So one way we can get around this is to do multiple random searches, and if they're all converging on the same basic alignment, maybe even an identical alignment, then we can be more confident that we found a good approximation or a viable alignment. Now, in addition to just these basic types of alignment methods, there are other things that we might want to consider, like maybe some information. If we know something about the gene itself, we may want to incorporate that into our how we are evaluating the tree score. Ribosomal genes are an interesting one because ribosomal genes have this secondary and even tertiary structure where they fold over into a three-dimensional shape. And by evaluating that, we might have some input and inference. So for instance, this site is going to be connected to that site because they form this stem structure. And so we know that there's going to be some input that if this site mutates, we might expect the other one to have some sort of a compensatory change. And so we might implement something about the three-dimensional or two-dimensional structure. If we're looking at a protein coding gene, then we might want to implement something about the position of each of these nucleotides. Is it at a first position versus a second position? and how does that impact the evolution or the changes that we might expect to see. So we can also implement information about the genes themselves to help me find a more optimal alignment. Now again, that's just kind of a big picture view. There's a little bit more in depth that we don't need to know in the paper. And if you were to get into this, if you make this, say, the subject of a PhD, which, we're getting, and that's the way PhDs are, right? They're usually very, very specialized. Multiple sequence alignment is a very specialized discipline. And if you were to get into it, you'd get even more and more in depth into maybe even the mathematics and the theory behind it. But lucky for most of you, maybe even all of you, because that's not your educational goal, this is the detail that you need to know. So review those different types, know a basic outline of how they work, and be able to compare and contrast those different